I'm here to talk to you guys about Goodeids. It's a group of fish that I didn't hear about until I was much older, and then it just sparked something in me. My lovely fiance is walking around, and she's obviously one to thank for allowing me to do the things that I do. So here's my presentation. Goodeids, background, classification, and interesting factoids. So if anyone does watch my videos, you know I like to ramble a little bit, and I digress and do a lot of background info. I love to talk about the history, the etymology, all kinds of stuff. So let's get into it. If anyone knows Chris, I've gotten quite a few fish from him, and somebody that I can bounce a bunch of messages off as well. And then Greg Sage, who was actually my first Goodeid pen pal. When I first found out about them one late night when I was surfing the internet looking at rare livebearers. And also a special thanks to my mom, who's back there with some of the kids. letting me keep all these animals when I was a kid, and again to my fiance for letting me do this now. My professional career is in science, primarily in microbiology and biochemistry. I've been keeping aquariums and such for over 20 years. I'm not that old, despite the white in my beard, so that's most of my life. I didn't even hear about Goodeids until I was in my 30s. So that's one of the reasons why I was so fascinated by them, is because they're obscure, they have a very interesting life cycle, the rarity of them, and so many of them are just so beautiful. I couldn't believe that more people hadn't heard about them. If you walk into any pet store, you ask them about Goodeids, you're going to get a sideways look. Like, pretty much no one's heard about them, unless you go to a place in Michigan out by Kingston. I do need to plug my YouTube channel, so if anyone is not subscribed, I do post regularly about a lot of the fish down there. It's not all Goodeids, but it's probably 98% Goodeids. Because I love the classification of all these things. If you know how everything breaks down, you have a better idea of how that fish or how that animal really works. And also, if you do watch my videos, I love to say the scientific names. I love it. So, I've been calling this Anna of Turbofish because I don't like to pronounce the P in the Greek words. Like when you say pterodactyl, you don't say pterodactyl, it's pterodactyl. And it comes from pteris, which means wings. So, great fin fish, which is the projectile mouth parts, telescoping, which is most of the fish that we probably keep. Mobile premaxilla, Characodontini, which is that has there's Alan here actually brought me in an Aphanius species. That's a Characodontini from the Middle East. I'm not sure exactly. I still need to learn more about them. But this also includes things like the Factoids, the Chapalichthyini. A lot of the killifish are in this group, in addition to pupfish. So they all like. I've been catching these sorts of things, the Factoids, since I was little. So it's just one of those things that always attracted me. The superfamily Fungiloidea. That includes all of the fungulates. So we have our local fauna includes fungulates. So a cousin, which I'll get into in a little bit. And then finally you get down to the family Goodeidae. So within the family, there's actually two subfamilies. The Untitled is the oviparous springfish. So you have the Characodontini and the Factoids. And then the actual subfamily Goodeidae, where we have our livebearers. So, what exactly is a Goodeid? Again, I love the etymology. It's named for the American archaeologist George Brown Good, who he was working out at a Smithsonian in the late 19th century. A lot of different fish are named for him, including the fungulid Goodeids, which is the bluefin killie that you can find out in like the East Coast. There's a lot of other things. David Sir Jordan, who's another ichthyologist from the golden age of ichthyology, he named the genus Goodea for George Brown Good, and then ultimately Goodeids were named for him later. The family arose in the Miocene with the Goodeidae, the livebearers, diverging sometime around 17 million years ago. The oldest fossil that they have is actually this Chapalichthyini occidentalis, which was actually, they would have placed it in the Chapalichthyini tribe. This picture is actually from the publication. I got this one from Michael Cook. Every other picture that's in this presentation I took. It's mostly fish from my basement. There's really just one slide that has some fungulates that I caught that I thought were pretty cool. So that's the disclaimer. Everything else is from my basement. Alright, so commonly referred to as split fins. My simple description for them, mostly live-bearing North American tooth carps with special skulls and modified anal fins. Yeah, it's the best way to describe it. The more elaborate description, Goodeidae is a family of fish within the order Citronodontiformes, differentiated from close relatives fungulates and the profungulates, which is another Mexican killifish, by the following typical characters. On the anal fins, the radials are fused, and then there's a couple elements of the skull that are in common, but really, like, they kind of all just look a little bit similar. Within this family, then the Trichinae, the egg layers, they're all found in Nevada, and the Goodeidae, the live bears, are actually down in central Mexico. Again, to talk about the variability, you have herbivores, carnivores, and omnivores. You can pretty much find, they'll eat just about anything, but I do like to read the species descriptions before I really start speaking. Like a too specific of a diet. Most of them are small and perfectly suited for the aquarium, between like 2 and 4 inches, but some of them, like the Scipia francese, Neotoca bilineata, they'll only get to about like 1.6 inches for a max size, which for like, if you were to look at like Scipia multipunctata, it gets closer to like a 3. And I have some real big multipunctata females, and I know there's been discussion about how differentiated the genus Scipia or the multipunctata and the francese are. I really think that the multipunctata is just a much larger fish, in addition to having some black spotting. The largest, so actually this up here, this is Neotoca bilineata, the 2-line Scipia. The largest are the Goodea atropinus, which can get upward like close to 8 inches, but also Allophorus robustus, the bulldog Goodea, it only gets to about like 6 inches, but it'll get way thicker. I'll show it later, but I had seen some of Meek's specimens at the Field Museum, and one of the jars that had Allophorus robustus, there was one fish in the jar. It filled it. It was impressive.
Yeah, Morelli just got a 14 inch.
That's ridiculous. Yeah, it is kind of. It's not. This is a bulldog idiot. It's not the prettiest, but this is just like a baby. I got that fish back in December. They're already twice the size they were, and they eat 3 times a day all the time. That's great. I had mentioned the Goodeiid cousins in the local fauna. Although both Goodeidae and the Bacillid families are known for live bearing, that's just an example of convergence. So to talk about like live bears is kind of like a paraphyletic term. The best way to describe paraphyly for me is when I think about trees. All the deciduous trees and flowering trees, they are more closely related to like a tulip than they are necessarily to like the pine tree, the gymnosperms. So that's trees is a paraphyletic term. There's a lot of things that are like trees that are tree-like, but they're not all closely related. So, I don't know. I really get down with all these terms. So, like I said, it's an example of convergence. The Goodeids are actually more closely related to the Fungulates. And locally, we have the starhead top minnow and the black-striped top minnow. And I've caught those in Michigan and in Illinois as well. So it's really nice to find that these things are around us. So it's the closest thing we have to Goodeids, like in Illinois, other than my basement. The Factoids are actually more closely related to the Anableps, which would be like the Four Eyes, and even the Genencia is in that same family. Girardinichthyini, they get colored. The males have color on their pelvic fins, so it's like an opposite evolutionary way to go. Just completely drop it. Most of them are federally protected, but you can own Goodeides Baileyii Moape, which I had gotten these from the ALA auction last year, and they're beautiful, very tiny. There is genetic evidence that was presented by Kyle Pillar's lab. I want to say it was in 2017, which dwarfs a redescription of the fish within this subfamily. The Embotrichthyes, they only have the one fish, so it's not, they don't have a whole lot of genetic divergence evidence, but the Goodeides nevadae is probably going to be split into two species. And then there was formerly 5 subspecies of Baileyii that are all actually going to be sort of done away with, because it actually only supports, the genetic evidence supports 4 species. So, interesting. But that's a story for another time, because I'm talking about livebearers today. Subfamily, Goodeidae, the livebearers. The distinguishing traits: males have a notched anal fin called the Antipodes. This is a young Xenotoca melanosoma, and I zoomed in on his notched anal fin. It looks like a mitten with the thumb pointing forward. And that's the best, really the best way to tell for really any of them, because the other distinguishing characters for the subfamily are the Trophotania that the babies are born with, and then also that the babies before they're born have a really small yolk sac. I talked a lot about this in the article I submitted for the ALA. It acts as an internal, an external intestine for the developing embryo. So it grows this stuff out of it, and inside the back of the mother's flank area, she secretes a fluid, and these sort of form like a junction point and absorb the nutrients up and carries the blood vessel through the blood vessels back into the baby, and it connects right near the anal opening on the baby. So this right here, this is the Xenotoca doadrioi. I actually brought a bag for the auction of the doadrioi. It was really cool to be able to get these photos when those babies were born. Down here, this is Neotoca bilineata. It's like just a handful of them. I really thought it would turn out better in this, but you can see that they have the Trophotania hanging down. It looks kind of like a white octopus. With Goodeids, they refer to this as matrophic viviparity. So the mother provides the nutrition for the developing embryos. And I went over like a few different other types of information. With like a lot of the cichlids, with there being an external fertilization, they refer to that as ovuliparous. An internal fertilization with egg laying, which I think there's some facilities from Africa that do that. I'm not 100% sure about. They're not live bearing. There are, Alan would know. But that's what a chicken would do. A chicken is oviparous. And then an internal fertilization where the embryos feed on yolk and then are born alive would be called ovoviparous. Most of the livebearers, most of the live-bearing bacillus, things like guppies and swordtails, and like the lamia, those guys are ovoviparous. But this guy down here, Heterandria formosa, the least killing fish, is actually a matrophic bacillid, which is really interesting, and there's not a whole lot of them like that. So it's kind of an outlier, but I thought it was a really cool fish. I got this from Steve. So if you want least killifish, Heterandria formosa, seeds are great. Alright. Continuing on with this, my trophic viviparity. The fertilization is always internal. There is no sperm storage. Every pregnancy comes from a separate fertilization event, just like people. And gestation is typically around 2 months. There's another bilineata up there. You can see the eyeballs in the back. It looks pretty similar to the pregnancies of bacillids, to be honest. This is Girardinichthyini multiradiatus with the little baby sticking out the back. Live birth. The fully formed fish are born alive and swim, but they tend, for me, to rest on a lot of the plants up near the surface. Tends to be a smaller brood size. Younger fish, younger females, when they're born or when they have babies, it'll probably end up being like a smaller. You might only end up with 3 to 5 babies with a young one. And it seems also like the youngest ones, the babies, don't get a chance to really develop all the way, so you'll get like crimped tails and things like that. But some of the older ones will produce much larger. I have a tequila fry drop that just happened recently. That was 19, and that was my best tequila drop, so I was pretty happy with that. Additionally, the fry tend to be a lot larger than the bacillid fry. This right here, this is Aniobius toweri, the blue-tail Goodeids. And those fry are close to 1 inch long, honestly, when they're born. And it's really surprising because the fish that I had that had those babies that were close to 1 inch, they were only like 2.5 inches at the time, so I couldn't believe that they were all folded up in there. Sexing males and females, really the best way is to just find, again, that the andropodium. This up here, this is Chapalichthyini. They do not have much dichromism. When you look at them morphologically, the males will have a little bit longer dorsal fin, but the coloring is basically identical. With a lot of the other species, like especially the Scipia, you'll see the more exaggerated the finnage and the colors, the more competition that there ends up being from mating. I also have, like I showed the picture of the Lamia. The Lamia tigers, those guys, they refer to them as sneak breeders or ambush breeders. They do not display for the females at all, and the females and the males look basically identical, aside from the gonopodium. I really liked these pictures and I wanted to put them in this presentation. This is the Scipia multipunctata, Lago dei Camiforo. This is the line that Pat maintains. And you can see the male with the black spotting. Multipunctata actually means many spots. With a golden color towards the flanks. The females look very different. They have no notches on the dorsal or the anal. And then it's basically just a tan fish with some spotting along the lateral line. So I forget exactly which species it was. It might have been Xenotoca variata, but when it was originally described, the guy had only females from one, and then he had another sample that had males and females, and he described them as 2 different species. Within Goodeidae, they're broken up into certain tribes. There's always a need to kind of group things by the way they're most related. The Characodontini is the oldest group, so that's the group that that tribe contains Lateralis and the Audax. Ilidontini is the next oldest, but the genetic evidence that that they were referring to before that broke up Chapalichthyini and the Girardinichthyini tribes is not necessarily supported. I don't know if anyone else saw the most recent Kyle Fuller paper. They were trying to establish homophily for the different tribes, and it wasn't fully supported with the Girardinichthyini. Some of the Scipia tend to be more closely related genetically to the Characodontins than they are to Allotopa. So that paper is still new. I had talked with Michael about it. I'm not necessarily sure of how to go from there. So just grouping them. This is the organization that's basically supported by Omar Dominguez from the 2010 publication. This is the Loberos lateralis up there, the top right. That is the Los Pinos audax. This is the El Toboso Audax Black Prince, and then down there is the Puente Pino Suarez. That one is also Audax. With this one, there are only the 2 species. There was a third one, Garmani, which that was described by David Starr Jordan in the late 19th century. But I think that species, I had literally just talked to Alan about this. I think that was described because of maybe more so an American desire to describe the fish, because Lateralis was described by a Swedish guy working in the British Museum in 1866 from specimens. He didn't collect anything, and Jordan actually collected stuff. So neither here nor there, I guess. In 1986, when it was formally described, the Black Prince, that was the only Audax location, but I think it might be the 2010 Omar Dominguez paper, or it could have been one of the other subsequent discussions. But the Goodeids Working Group, their website actually includes these other 3. Those are all as Audax, so it's been expanded. And that's based off of a break in their territory, where it's called the El Salto Falls. So the fish that are above the falls are referred to as Audax, and the fish below would be Lateralis. So I don't have the Amato Nervo, but that one I think is a true Lateralis. And then the Guadalupe Aguilara, I think that one actually is the Audax. So that's the way it's split. So I may have referred to some of the other ones in older videos as Lateralis, but now I follow based on the distinction from the falls. And this is the next tribe, the Girardinichthyini, the next oldest group. Up here I have a whitey eye. This is actually a baby hanging out. Xenotinia resolane. This is an Alodopticus Tamazula, and then an Iliodon persistens. There's the 3 genera in this group. They're all basically like tubular fish. They look kind of like sausages if you look at them straight on. This is the Chapalichthyini tribe, which is probably one of the more numerous in terms of the species and the genera included. The Chapalichthyini, this is Chapalichthyini encostus, the namesake for the tribe. That fish is named for the lake that it's found around. It's pretty much all found in the lake. Encaustus actually means burnt because it looks like it's been grilled, which is apt because it was also being used as food by the people that lived on Lake Chapal. So up there, that's a newer picture of my robustus. It's much bigger. That's the bulldog Goodeids. That would be I think technically, because of the way they were being organized, the Goodea group, or the Goodea genus, actually becomes the stem fish for the Chapalichthyini tribe. And then they placed Haurai at the stem of the Girardinichthyini group. But like I said, that the monophyly of those groups isn't well supported. So they kind of that's why they're kind of all together. But I didn't include it in there because it's still of uncertain distinction. But up here, Chapalichthyini encostus, this is the Xenotogodroid with the banding. This is Zoogeneticus porpacius. And again, the Alopterus robustus, which the location I have for that is Lago de Zacapu. So, very cool creature. Oh, before I go on, with this group, I had mentioned that the Iliodontiini are more tubular. These guys are more flattened. And you'll see a lot thicker bodies, especially with that big Alright, and then in here, this is the Girardinichthyini tribe, which the way it was being considered, I think, after Turner and I think the Miller papers, was that it was the most derived of the group and the youngest tribe. This is the Scipio multipunctata Tangansicuro. I actually bought this fish too for the auction. Over here, that's the Sayula Scipio. That's actually an undescribed species. Girardinichthyini multiradiatus, golden sailfin, or the dark edge splitfin. Of note about this one, because I think Pat would be upset if I didn't say anything. This is a cold water species. You can't let it get above 70, otherwise you'll kill them. So I have it on my floor in the basement. Yeah, and they're doing great. And then over here, this is Goodeids, the bumblebee Goodeids. That's the Rancho El Molino that Rick had collected in 2014. And the cool thing about them is that there's a golden form and a black form, and you can see down here, this male there is more of the golden form, whereas the more jet black body, that one seems to be proliferate more so in captivity. 1769, that was when they first found that there were these live-bearing fish down in Mexico. It was being reported back. But it wasn't formally described until 1837, and it was Girardinichthyini viviparus, and that's Xochimilco is the location on that fish up there. I have a female that's very ready to drop, and I'm very excited for that. She's huge. The interesting things about that, when it was first described, it was so common. They used to find it in all the ditches all around Mexico City, and it was a food source for the poor. So it was that common. Now it's critically endangered. And in fact, a lot of the wild populations are riddled with parasites and found in heavily polluted areas. But because of the understanding that people had of classifications and how that all worked, it wasn't as advanced as it is today. It was originally described as Cyprinus viviparus, which is actually the genera that includes carp. So that's what they thought of it as at that time. I had mentioned Characodontini lateralis was described by Albert Gunther in 1866. The Characodontins, before I think it was all brought together, I think by like Reagan or potentially Meek, it was the Characodontini was a whole family, and then you had Goodeids separate. So that's why I thought it was weird that they based all of this off of fish specimens that they had examined in a museum way far away from where they were collected. I don't even know that they had good collection information at all. In the 1870s, 1880s, George Brown Good, the namesake for the family, was working at the Smithsonian and at the U.S. Fish Commission, and David Starr Jordan was working at Indiana University. Spencer Baird, who was George Brown Good's colleague, hired David Starr Jordan to do surveys out west, so there was a connection there. And then in the 1880s, Seth Eugene Meek, he went to Indiana University, and he was studying under David Starr Jordan, and he did collecting with Charles Henry Gilbert. I'll bring it around, okay? I'll bring it around. So the history of study. Meek and the Chicago connection. Seth Eugene Meek, he actually worked at as the assistant curator at the Field Museum. And he was the first compiler of comprehensive books on the fish of Mexico. And he did 2 trips, 1901 and 1903. And up here, that's actually the Aloperis robustus that filled up an entire jar. And I actually convinced my fiance to go with me to see these specimens. So she's a saint. So, yeah, we went 1901, 1903. And those publications, they weren't just on Goodeids or just Bacillids or just livebearers and things like that. It was on everything. It was a very comprehensive study. And in those 2 books, they're actually, or the 2 publications, described 5 different species, which I have them listed here. The genus Scyphia, that was actually named for Meek's boss, Frederick James Volney-Skiff, who was the first director of the Field Museum after it opened following the Columbian Exposition. So I thought that was really cool, and I wanted to say that for the Chicago Livebearer Scoiety, that there was such a connection to Chicago that you wouldn't even really think about. And it was funny messaging the guy at the Field Museum about seeing the Goodeids specimens. Like, okay, that's fine. So he let me pick them up and everything. This is Characodontini garmani. is technically only known from a single specimen that was collected by David Starr Jordan and Jordan Everman in 1898. So it makes sense that when Meek went, because he had worked for David Starr Jordan and studied under him, that he would base his identification of these specimens on the fish that his mentor had described. So that's not a whole jar of Characodontini garmani. Those are actually lateralis. And then down here, this is the Scipia lermae, and that's the fish that Meek erected the genus Scipia, which is one of my favorites because they're beautiful. So that genus was erected because of this fish here, the Scipia lermae, that were collected in that jar, which I think is awesome. In the 1910s, 1960s, Hubb's studies, he actually worked at the Field Museum as well. I had some pictures of his specimens, but they didn't make the cut for the, I'm sorry. But then Carl Hubbs, when he was at Michigan, Robert Rush Miller, that everyone's familiar with, if you have Amicus splendens or the Xenotania resolanae from Pat, those are all Miller collection. There's a posting that he had done, or he had posted on Facebook all the different Miller collection information. I saved it, so if anyone has questions about that after, I can share it, or I can just give you Pat's email and you can bother him. So at Michigan, Robert Rush Miller studied under Hubbs. He married Hubb's daughter, and then he goes on to become one of the most prominent ichthyologists in the study of fish in Mexico, which that connection, David Starr Jordan, Robert Rush Miller, that is basically most of the Goodeids species that are described. And it was just like this little group of people just passing on their love of Goodeids, which I think is cool. As I had mentioned, I love the etymology, and I harp on the Linnaean binomials. I think it's important to know them, and also to recognize that the names they're not scary. It breaks down, and each part actually means something. So they're not as fun as a lot of the facilid names, which I actually had to edit something out of one of my last videos because it wasn't quite kid-friendly, because I was asking Pat about the xenophalis. Xenophalis translates to strange male part. The name Priapictes, Priapus was a Greco-Roman deity of fertility, and there's a real famous story about him challenging a donkey to something. You can look it up. This stuff is all out there. He's also, Priapus was in a famous mosaic that was in Pompeii, where he's measuring it against gold. He's weighing it against gold. Okay. So that's his fish. The Xenophalis strain. This is Factoids. Literally is just, yeah. I don't want to say it out loud because my friend is recording this for me. And then, so not as fun, but the names still all mean something, and I think it's fascinating. Adniobius toweri, which that name right there, Adniobius, it looks daunting to see it, but when you break it down, the A means without, and you see that like asymptomatic for the doctor, right? Without symptoms for that. So the tania, that refers to the Trophotania, which we talked about earlier. And then the bias, it literally translates to without a life band. And then toweri is an honorific epithet because the fish was actually discovered by Dr. William Tower at the University of Chicago. So, and that was described by me in Chicago. Situation in the wild. I know when we had Dr. John Lyons come, he talked quite a bit about them being imperiled and the reintroduction efforts. So I didn't want to hit on that too much because I felt like we had just done it. But most of the species are imperiled. Primarily due to pollution, irrigation, use of the fish as food. As I'd mentioned, the viviparous used to be found like so commonly that they were just basically giving it as food to the poor. Now it's almost gone. And then the introduction of non-native species that'll either compete with or just eat them. So black bass, there's lots of reports of the Pseudoxiphophorus bimaculata. Xiphoporus helleri, green swords, domestic strains, all kinds of stuff just taking over and out-competing the Goodeids. So of those 42 extant species, 37 are listed on the CARES priority list. And of those 5, 2 of them are federally protected and you can't own them as a pet, so they wouldn't be listed regardless. So that leaves only 3 of the 42 species that are not considered to be on the CARES priority list. This over here, this is actually Scipia francese. I had a couple nice shots before. The CARES fish, it is an acronym, but it doesn't quite line up. You can see the words right there. There's an extra E and an extra S at the end. But it's designed to be implemented in the in aquarium clubs. You can assign a coordinator. They changed up so people can now register their fish independently of being in a club. It remains to be seen how well all that works out, but this is the website. They give classifications for the fish based off of either the IUCN or published population studies. So there's citing of, like, if you go on the priority list, the source for those listings, it says JL, that's John Lyons, who's the head of the NAGWG, the North American Goodeid Working Group. So 37 of the 42 species are listed on this, ranging from vulnerable to extinct in the wild, the francese being extinct in the wild. I have one fish that is registered for CARES, and those are the exact pictures that I used for my submission. So I thought that was fun, and it's also the species that I brought here. Most of my tanks are just ambient. In the winter, it actually gets real cold down there. So it was up in the high 50s for a little bit when we had that polar vortex. I have a lot of floating plants. It's either the dwarf lily, or I do a lot of guppy grass. I recently got some hornwort that I'm excited about. I have a simple hang-on back filter, and I put a sponge over the filter inlet so babies don't get sucked up. I don't have anything automated, and I have LEDs because they're low wattage and they last for years. As opposed to the old T5 ballast and stuff that I used to use, which consumes a lot of energy. The tanks now, like the entire wattage to run some of these tanks is less than like 50 watts, which is, I think, is great. And this down here, like the babies, when they're born, oftentimes they'll hang out right near the surface. So if you have something like this is a dwarf lily, they will hang out on that. It doesn't form like a full lily pad. It hangs out just underneath. In the species description for Anaeobius tauri, it references something similar to this. And when I talked to Michael Cook about it, he said that was the perfect plant to grow for breeding Goodeids. So if you can get this dwarf lily in like a 20-gallon tank with a small pot, it'll grow right up near the surface. You can just have tons of babies. That tank I had like 80 tauri in, and that's a big fish in a 20-gallon. With my fry, I tend to pull most of them. I had posted some pictures where it's just those plastic storage bins with air stones to keep the water moving a little bit, and a lot of floating plants. And it's not so much that the parents are going to predate on the fry. It has more so to do with the fact that the parents will eat all the food, and the babies will just not get anything. So if you want to do survival of the fittest, if your colony's huge, like I've seen some of Pat's tanks, his colonies are amazing. Mine are not as amazing, so I need to optimize my fry survival rate. So I have a bunch of them. So those are my adult Altogodigazii, and then here's some babies in my fry tank. So, and as I had mentioned, like with the survival of the fittest, once you have like a decent number, multi-generational colony, you can just let them colony breed, and you don't necessarily have to pull anything, and you won't see too much in terms of like any fry predation. And then they end up getting all. It's a beautiful sight when you have a ton of Goodeids in a tank, and then you go to feed them. Mind-blowing. So talking about feeding, like I had said before, they eat all kinds of different stuff. This top one, Amicus clemens. This one down here, that's Xenophorus captivus, the relic split fin, which I'm pretty excited to have. I have gotten fried from that. I heard somebody say that it's just an ugly green fish. I think it's super cool. It gets a little bit of an iridescence. It looks kind of like a mini jeweled split fin. Which is funny because with the genus Xenotoca, most of the Xenotoca are actually more closely related to the Xenophorus, and then the Xenotoca variata that I just said that it looks similar to is more distinct. And that would actually be the true Xenotoca. Everything else should be reclassified. But anyway, I just want to make sure that if you do get Goodeids, that it's a good idea to research the species because there is so much variability. I had mentioned keeping those tanks ambient. I do have two tanks that I heat, my Crinictes baileyii and also the Adniobius toweri. I heat those to about 76. Everything else is probably in the high 60s. And they do well in groups. So many times, like, if you hear somebody say, like, oh, their tank's overcrowded, when it comes to these guys, don't worry about it. If you have a lot of plants and everything, they'll hang out in the open areas and they'll be right on top of each other. That's, again, the Scyphia that I brought for the swap. And I love that picture. That was just a amazing picture. And down here is some Zoogoneticus tequila, which is, I needed to include more pictures of tequilas because it's actually in our logo for the Chicago Livebearer Scoiety. So, with these, if anyone, if you're on the fence about keeping Goodeids, or if you have a situation where you keep multiple tanks and you have some space, why not dedicate just one tank to keeping one of these internal fish going? There's really not that many keepers. I posted about like a bunch of the different species, and someone was saying like you don't really see this anymore because so many of them are disappearing. There's a handful of species in the Alitoca genus that I really only know of one person that's keeping them. And he's looking for like a male Alitoca goslini because he just has 4 females and that's it. And we don't know anyone else other than potentially someone in Europe. So if they are available, I highly suggest picking them up. And thank you to those that already do their part to maintain the species, especially Pat. Right? All right. So thank you for coming. Are there any questions?